Hi, I'm Karen, and today I will present over collagen and its role in the extracellular matrix. To start off, I should just point out by saying that collagen is the most abundant protein found in mammals. As many as 28 different types of collagens have been categorized based on the combinations of alpha chains they contain. These various collagens were classified with Roman numerals from 1 to 28 based on the chronological order of their discovery. I have listed which ones belong to which group, but regardless of what type of collagen it is, they play a big role in the integrity of both dense and connective tissues found in the body. So, the structure of collagen molecules consists of three polypeptides that are known as alpha chains. The alpha chains intertwine, forming a right handed triple helix, and every third amino acid in the chain is a glycine molecule, with the exception of alpha chain ends. Either a hydroxyproline or hydroxylysine usually precedes each glycine in the chain, and a proline frequently follows each glycine in the chain. Along with proline and hydroxyproline, the amino acid glycine is essential for the triple helix conformation. Sugar groups are associated with the helix, which are joined by hydroxylysyl residues. Because of these sugar groups, collagen can be also considered a glycoprotein. A collagen molecule may be homotrimeric or heterotrimeric, meaning either it can consist of three identical alpha chains or the molecule can consist of two to three genetically distinct alpha chains. In a basic explanation on the synthesis of a collagen molecule, we first have the formation of mRNA in the nucleus that results in the synthesis of pro alpha chains that are initiated by signal sequences and ribosomes on the RER. In the RER, glycosylation of hydroxylysyl residues and cleavage of the signal from the pro alpha chain also occurs. Pro-collagen triple helix molecules form from C terminus to N terminus um, directions. So the triple helix is stabilized by chaperone proteins and the formation of intra and inter chain hydrogen and disulfide bonds. A transport vesicle then takes procollagen molecules to the Golgi apparatus. As we have learned recently in class, the Golgi packages the procollagen into secretory vesicles for transport towards the plasma membrane. Vesicle movement is assisted by microtubule associated motor proteins. After exocytosis, cleavage of trimeric globular C and helical N procollagen domains by proteinases leads to polymerization of collagen molecules into collagen fibrils. As I've mentioned before, collagen is the most abundant protein in the body. And with that being said, collagen is important to be dispersed all over the body so it can provide that structural integrity and that support and filtration barrier. And it provides that resistance to force, tension, stretch, and pressure. Aside from that, it facilitates the movement of endothelial cells that you find in, for example, the kidney, or it also contributes to bone mineralization by forming lattices. And diseases that are associated with a malfunction of the production of this protein is osteogenesis imperfecta. And it's basically a genetic defect, and it affects the body by not having the ability to make strong bones. Either a person has too little type 1 collagen or the poor quality of the type 1 collagen due to a mutation in the gene um, can really mess with the framework that the bones are formed around. Um, the result in these cases is usually fragile bones that break easily. And although not always, it's possible to diagnose osteogenesis imperfecta just based on clinical features. There are many types of osteogenesis imperfecta, but in type 1, the deficiency mostly arises from an amino acid substitution of glycine, and it puts in a bulkier amino acid in the triple helix structure. And as I've mentioned before, glycine is just an important factor in collagen architecture. Generally, we can see that in a patient, they can have repeated fractures after a minor trauma, brittle bones, abnormal teeth, thin skin, weak tendons, blue sclera, and progressive hearing loss. Moreover, in a paper published this year, presented a study that used metastatic non small lung cancer cells, and they tested out different concentrations of collagen type 1 in the presence of transforming growth factor beta, and to see how the conditions of the extracellular matrix plays a role in the dissemination of cancer cells and how it contributes to the first steps of tumor colonization. From what they were able to conclude from the study was that those cells that grew in enriched collagen matrices or matrices migrated more slowly and tended to form cell clusters spontaneously, meaning that cell aggregation and cluster stability was enhanced when they were in a denser matrix environment. And in this paper, they highlighted also how TGF beta inhibition reduced the formation of bone metastasis, and how TGF beta seemed to have a relation to the activation and progression of disseminated tumors. So now I ask you, w- what induces bone metastasis formation? In other words, what induces TGF beta concentrations and through what signaling? I'll leave the link of the paper in the description so you can go ahead and go a little more in depth in this investigation. That's it for today, guys. Thanks so much for listening to my presentation. Bye.